Hello students, I am Ashish Badiye, Assistant Professor at the Government Institute of Forensic Science, Nagpur. Today, I bring you an exciting and learning episode in BSc Forensic Science on an important unit, that is Fingerprints Part 2, Anatomy of Skin and Fingerprints, in which we will discuss about the general anatomy of the skin, the various layers of skin, formation of fingerprints along with the role of genetics in it, the composition of fingerprints, etc. We will also learn about a few rare conditions where fingerprints may be absent. We will wind it up with a brief about the important characters of fingerprint that make them useful for forensic purposes. Let's begin our episode with a glance at what we are going to learn today. Module 1, General Anatomy of Skin. Module 2, Formation of Fingerprints. Module 3, The Role of Genetics in the Development of Fingerprints. Module 4, Composition of friction-rich skin. Module 5, Rare conditions where fingerprints may be absent. Module 1, General Anatomy of Skin. The skin is an organ composed of three anatomical layers, namely epidermis, dermis and hypodermis. These anatomical layers together function to provide the body with a protective barrier, body temperature regulation, sensation and excretion. The outer layer of skin is the epidermis. The epidermis prevents water loss through evaporation, acts as a receptor organ and provides a protective barrier for the underlying tissues. Melanocytes, the pigment producing cells of the epidermis, play a key role in the protective barrier. The pigmentation produced by the melanocytes shields the DNA of the keratinocytes from the sun's harmful rays. Additionally, the melanocytes are responsible for the synthesis of vitamin D. The dermis is a layer of connective tissue that supports the epidermis. It is a network of cells, fibers, blood vessels and gelatinous material that provides structural support and nourishment for the epidermis. The dermis serves as a blood reserve and participates in sensory reception and temperature regulation. The hypodermis lies under the dermis and is a loose connective tissue that contains a pad of adipose cells that counter the body and serves as an energy reserve. Fibers link the epidermis to the dermis and the dermis to the hypodermis. The only skin appendage of the friction-rich skin is the eccrine sweat gland. Although sweat glands are distributed over almost the entire skin surface, the friction-rich skin has the highest concentration of eccrine glands, 2500 to 3000 per 2.5 cm square. The sweat glands of the friction-rich skin are also the largest on the body. Eccrine sweat glands participate in temperature regulation by secreting sweat and assist in the excretion of metabolic waste. The skin can be a good indicator of health. A person who is in shock may have pale skin and goosebumps, and someone with a fever may feel warm to touch. This basic information is a common knowledge. Being a forensic student, as our habit should be, let us go a bit deeper and get beneath the skin of this topic. Epidermis. This tough layer of cells is the outermost layer of skin. It gets its toughness from a protein called keratin. The epidermis has 5 layers. Stratum corneum is made up of dead, mature skin cells called keratinocytes. These cells are constantly shed and replaced by cells from the lower layers of the epidermis. These cells have lost most of their internal structure and organelles. Stratum lucidum is found in thicker skin and helps reduce friction between the stratum corneum and the stratum granulosum. It is composed of dead, flattened cells. The skin is thicker in some areas like the soles of your feet and thinner in others. Women also tend to have thinner skin than men do. Stratum granulosum is where keratin is formed. The cells in this layer also produce materials that prevent evaporation, which helps waterproof the skin. Stratum spinosum contains the keratin producing cells that were formed in the stratum basale. Keratin is a major structural component of the outer layers of skin. Stratum basale forms the deepest layer. The cells of this layer continuously divide and form new keratinocytes to replace the ones that are constantly shed. This layer also contains melanocytes, which are the cells that produce skin coloring. Dermis. This lower layer of the skin contains collagen and elastic fibers that give strength to the skin. This layer is also where the vasculature and nerves live. Together, the epidermis and dermis form the cutaneous layer. The subcutaneous layer lies underneath the cutaneous layer and is sometimes called the hypodermis or superficial fascia. It holds most of the body's fat, so it varies in thickness from one person to another. Creases form over joints because the skin always folds the same way as the joints bend. The skin is thinner in those areas and is firmly attached to the underlying structures by connective tissue. The integument also includes the structures that grow out of the skin plus a couple of glands. Hair. The protein keratin forms hair. Hair has an inner layer which contains pigments that give it color and an outer layer. It grows out of follicles, which are little pockets of epidermis in the dermis. The shape of the follicle determines whether hair is curly or straight. Each follicle contains a hair bulb from which the hair develops. Erector pili muscles connect the hair follicle to the skin. Nails. Keratin shows up again in the form of plates found on ends of the fingers and toes. Underneath each nail is a nail bed with a root at the proximal end. Sebaceous glands. These glands are connected to the hair follicles. They produce sebum, which is an oily substance that helps keep the hair flexible. Sweat glands. Sweat glands are coiled tubular glands found in most of the skin. The secretor portion of each gland lies in the fascia with a duct that runs up to the surface of the skin. The fascia is divided into two types, the superficial fascia and the deep fascia, which forms the following structures. Investing fascia. This part of the fascia covers deeper structures such as muscles and ligaments. Intermuscular septa. The septa divide muscles into various groups. Subserous fascia. This part of the fascia lies between the body walls such as the thoracic wall or abdominal wall and the membranes that line corresponding body cavities. Retinacula. These band-like structures hold tendon in place while joints move. Brucae. These fluid-filled sacs are made of fibrous tissue. They are lined with membranes and contain a bit of viscous fluid. They reduce friction between body tissues. Major brucae are located near joints between the tendons and bones. Dear students, I know that this would have sounded quite difficult, but it was an important piece of information that cannot be skipped. Having a strong base would eventually help you in the long run. If you find it difficult, I would suggest you read and listen to it a couple of times more. Also, try looking at some images and pictures as it may make things a bit easier to understand and remember. Let us begin Module 2, Formation of Fingerprints. Firstly, what is a fingerprint? A fingerprint is a series of ridges that create a pattern on our fingers and are also found in apes, monkeys, gorillas, etc. You can also find other prints on your feet and palms. Why, you may ask? A tissue called volar pad causes fingerprints to form, yet they can be found only on our fingers, palms and feet. When a fetus is inside the womb, the development and formation processes that the tiny body goes through include forming fingerprints. Ridges and wrinkles on tiny fingers and toes become identifying marks months before he or she is even born. The friction ridge patterns start to develop between the 5th and the 6th week of intrauterine life and are fully formed by the 21st week. The fingerprints are complete by the time he is 6 months old. Once those prints are in place, they are there to stay. During early embryonic development, that is 4 to 5 weeks, there is swelling of mesenchymal tissue on areas of the sole, palm and digits. These areas are known as volar pads. While a fetus is forming, the pads on the fingers and palms start to develop between weeks 6 to 10. The pads stop growing at about 10 weeks of development, but the hand continues to grow. The volar pads are then absorbed back into the hands, and as the pads shrink, the skin folds to produce the ridges of fingerprints. Our prints start to form when we are 10 weeks old till week 15. Ridges start to appear between the times when the volar pads start to get pulled in till it is fully absorbed. The timing of these first ridges is important. Volar pads control overall alignment or pattern of the prints. How absorbed the pads are determines much of the pattern. Much of the spacing and number of ridges are also controlled by that. That is how most of a fingerprint is formed. Yet all fingerprints are unique. The ridge patterns that make the fingerprints have a few contributing factors, as shown by various studies around the world. The genetic factors have a part in this, but the fetus's location in the womb and the density of the amniotic fluid also influences the ridge formation. The fetus's movements in the womb and how big and fast it grows also contribute to the design of the fingerprints. A key link in fingerprint formation is randomness. Randomness makes every print different. Without it, most likely many people would have the same fingerprints. Because of the multitude of contributing factors, it's no wonder you will never find the exact prints on two people. Even identical twins do not share the same fingerprints. Once the fingerprints have developed, their pattern will remain the same. Even if you get cut or other superficial skin damage on the fingerprint, it will grow back in the same fingerprint pattern. This is why fingerprint identification is considered such a reliable source of identification. Students, let's begin Module 3. The role of genetics in development of fingerprint. Every aspect of the growth and development of a single cell into a fully formed human is initiated by a genetic blueprint. The capacity to form friction ridges is inherent within the developing embryo. The patterns that these ridges form, however, are limited by nature and are defined by the fingerprint community as arches, loops, whorls, combinations and transitions of these basic patterns or lack of a pattern. Although genetics may direct when and where ridges will form by providing the blueprint for proteins, nature provides the boundaries for patterning through physical mechanisms. Proteins direct cellular activity by facilitating biochemical processes within the cell. These processes depend not only on the protein derived from the gene but also on the many other non-protein components of the cell such as sugars, lipids, hormones, inorganic elements, inorganic compounds and minerals. Additionally, the physical environment around and within cells including surface tension, electric charge and viscosity contributes to the way the cell functions. Genetic information directs cellular function, serves as a link between generations and influences an individual's appearance. Some aspects of appearance are similar for each individual of that species, that is, those characteristics that define the species. However, within the species, for each aspect of an individual's appearance, many genes and external factors affect the final outcome of physical appearance. The genes involved with a specific attribute like skin color produce the appropriate proteins which in turn react with each other and with the many non-genetic components of the cell in complex biochemical pathways during the growth and development of the fetus. These biochemical pathways proceed under the omnipresent influence of external factors. The ultimate example of the role of the environment in friction-rich formation is monozygotic twins who share identical genetic information and very similar intrauterine environments, but on many occasions have very different patterns. The role of genetics is currently understood by the indication that several main genes in conjugation with a number of modifying genes may be responsible for volar patterning, but it is well established that friction-rich patterning is also affected by the environment. Module 4. Composition of friction-rich skin. The friction-rich skin includes the following. The ridges and furrows on the surface of the friction-rich skin are firmly rooted in the dermis by primary ridges and secondary ridges. The figure illustrates the structure of friction-rich skin. The primary and secondary ridges are interlocked with the dermis to provide support and strength to the friction-rich skin. Additionally, sweat glands extend from the primary ridges and are anchored in the dermis or hypodermis. Epidermis. It is the outer layer of tough ridged skin bearing only sweat pores. It contains no vessel, nerve endings or sweat glands. It functions as an aid in gripping and thus directly comes in contact with an object. The epidermis is described as a stratified, continually renewing epithelium that exhibits progressive differentiation in a basal to superficial direction. In other words, the epidermis is a layered tissue that must constantly replace the cells leaving the surface. New cells are generated in the basal layer and pushed toward the surface. As the cells move toward the surface, they undergo sequential changes in chemical composition. The epidermis is composed of several different types of cells: keratinocytes, melanocytes, Langerhans cell and Merkel cells. Dermis. Dermis is the layer just below the epidermis and contains sweat glands, nerve endings. The dermis is the connective tissue that supports the epidermis and binds it to the hypodermis. The dermis is composed of two layers, the papillary layer and the reticular layer. The outer papillary layer is a loose connective tissue containing anchoring fibrils and numerous dermal cells. The anchoring fibrils secure the dermis to the epidermis via the basement membrane zone. The papillary layer of the dermis forms the dermal papillae, papillaceae. These are protuberances arranged on the surface of dermis. Stratum mucosum. This is the lower level of the epidermis in contact with the papillary layer. Sweat glands. Although the skin produces several appendages, the eccrine sweat gland is the only appendage of the friction-rich skin. Eccrine sweat glands are found all over the body surface and functions primarily in thermoregulation. These are also called the eccrine glands, which excrete perspiration through the sweat pores. Eccrine sweat glands are classified as simple tubular glands whose ducts open at the skin surface at the coiled secretory portion of the gland, which is embedded in the dermis or hypodermis, and the duct extends through the epidermis. The fluid secreted by the eccrine sweat gland is predominantly water. The remaining constituents of sweat include sodium chloride, potassium, ammonia, urea, lactate, uric acid, creatinine and creatine, amino acids, sugars, immunoglobulin A and at times small quantity of albumin. The amount and constituent of individual secretions are variable and individual glandular secretions also vary. Environmental conditions such as temperature and humidity, exercise or stress affect the rate of production of eccrine sweat in an individual. The composition also varies in relation to age, sex, medical condition and diet. Eventually, these factors influence the quality of the developed finger marks along the environmental conditions post-deposition. Let us begin Module 5. Conditions where fingerprint may be absent. A very rare medical condition, dermatoglyphia, is characterized by the absence of fingerprints. The affected person will have completely smooth fingertips, palms, toes and soles, but no other medical symptoms. People with Naegeli-Franceschetti-Jadson syndrome and dermatopathia pigmentosa reticularis, which are both forms of ectodermal dysplasia, also have no fingerprints. This is the rare genetic syndrome. The anti-cancer medication capecitabine may cause the loss of fingerprints. Subject suffering from open cuts or scars in any of the upper phalanges of the fingers might not have the prints of such fingers. Absence of fingerprints, congenital milia, is a very rare syndrome characterized mainly by the absence of fingerprints, webbed toes and milia. Milia are small white papules, especially on the face. This syndrome is inherited as an autosomal dominant trait. Fingerprints possesses following characters that make them useful for forensic purposes. This we will learn in the upcoming episode in further detail. First is uniqueness. The fingerprints from two different fingers are never identical. Second is permanence. The ridge patterns on the fingers never change during life of an individual, except rare cases of mutilation like leprosy. Universality. Almost every individual born with fingers bear fingerprints on it. Next is simplicity of recording. The fingerprints get recorded on any smooth surfaces even without the knowledge of the owner. Simplicity of classification. Large collection of fingerprints can be classified in such a way that each gets its unique position in files. Hence, these can be applied in forensic science for classification, comparison, identification and individualization. Students, I hope you have found this information very interesting and learning. So, till next time, keep reading. Thank you.